Did you know that despite our apparent progress in cancer treatments, all common human cancers remain as incurable and as deadly as they were 200 years ago, if not removed completely with surgery in the early stage of the disease? And that we continue using the same primitive and mutilating surgical procedures created by our ancestors centuries ago. Did you know that cancer became the leading cause of death in the world in 2014, killing now 20,000 men, women, and children every 24 hours? Did you know that in the United States alone, 1,600 people die from cancer every single day, and that close to 50% of those deaths involve children and men and women in their most productive years? Did you know that in the United States, cancer is the leading cause of death due to disease for all children between the ages of 4 and 14, and all adults except the elderly? Did you know that the number of deaths due to cancer has increased dramatically in the last 100 years in the U.S.? In 1900, there were 67 cancer deaths per 100,000 individuals. But despite our continued progress in medicine since then, the number of cancer deaths per capita has continued to increase decade after decade. By 2011, there were approximately 185 cancer deaths per the same 100,000 people, an increase of nearly 300%. In contrast, during the same 100 years, all other diseases that were prevalent in 1900 have decreased by more than 90%. Such is the case for infectious diseases, and close to 30% of cardiovascular diseases, which is the leading cause of death among the elderly, despite our growing, aging population. In 2011, cancer specialist Dr. Frank Orguello asked himself this question: is there something fundamentally wrong in the way we understand the biology of cancer cells, and consequently, in the way we treat cancer?
So I decided to erase everything I knew about cancer from my mind. I decided to start from scratch. I put together new concepts to understand the biology of cancer cells, and I published it as a book under the title Atavistic Metamorphosis, a new and logical explanation for the origin and biological nature of cancer. In 2011, the same year in which I published the book, I initiated a proof-of-concept clinical trial in which I have treated close to 100 cancer patients with terminal cancer.
It was Rudolf Virchow, the father of pathology, who in 1888 noted that the life cycles of pathogenic single-celled organisms, such as bacteria and cancer cells, were indistinguishable. However, Virchow erroneously believed that cancer cells have acquired their malignant traits from bacterial cells after being infected by them. A more logical explanation for the similitude between cancer cells and single-celled organisms came from the British cancer surgeon Herbert Lumley Snow.
It is to be remembered that the whole complex organism, which we term the body, is built up of minute cell elements, which were once, to all intents and purposes, protozoic animalculae, like the amoeba. Cancer is but a process of de-evolution, of reversion of cells to a primordial amoeba-form condition, in which they become parasites, or rather, autocytes. Herbert Lumley Snow.
Snow regarded cancer cells as having reverted to their evolutionary past thus becoming independent single-celled beings once again, and re-expressing their pathogenic traits and behavioral characteristics of their unicellular ancestors. This phenomenon of reversion to the past is known in biology or in evolutionary biology as atavism. Atavism comes from the word atavus, that means grandfather or ancestor. There are several atavisms which are common. For example, the reappearance of tails in some children, or the presence of multiple nipples following what is called the midline, just like all the other mammals. The reappearance of legs in snakes. The snakes are reptiles, and for some time they have legs, and so on. So the reversion or the re-expression of a past feature, evolutionary feature, in an organism is called atavism. What Snow believed was that cancer cells was a cellular atavism, a reversion of one of our differentiated cells back to its ancestral unicellular and independent form. I reasoned that if Herbert Snow's conclusion was correct, that cancer cells function in their pathogenic atavistic unicellular and independent form, at least as a working hypothesis, then those cells should rely on cellular functions and survival strategies analogous to those used by their ancestors, the pathogenic single-celled organisms. To test this hypothesis, I selected drugs which are currently in the market and used for the treatment of diseases caused by independent single-celled organisms, such as bacteria, protozoa, and fungi. We created a clinical trial and we tested off-label different combinations and dosages as a salvage therapy to patients with advanced cancers who were discharged from conventional therapies due to lack of response or progression and were otherwise untreatable. Briefly, let's review cancers treated with this new drug-based therapy that I refer to as Atavistic chemotherapy, mainly to distinguish it from traditional chemotherapy.
There is intellectual property covering this invention. Some important points to be mentioned are the following. 1. This treatment involves the use of 6 different drugs used in strategic steps. The drugs are generic. Example, no longer patent protected as their patents have expired. All are FDA approved for use in humans and are available worldwide. 2. None of these drugs are used to treat cancer, but rather used for diseases caused by other cell types, bacteria, fungi, and protozoa. Some are not antimicrobials, but are needed to achieve the desired effect. 3. While these drugs are widely known, we now have a patent pending status for each of them, claiming a new, non-obvious use, drug repurposing. In addition, we have a robust patent claim involving a novel drug combination and formulation in order to achieve a non-obvious effect. 4. You as a pharmaceutical company or cancer organization licensing this invention would have full patent protection and could brand and market these new drug combinations for the treatment of cancer once all regulatory issues are fulfilled. 5. To ensure your success, Dr. Frank Arguello will give you 6 to 12 months of due diligence in order to carry out a compassionate clinical trial. Although this treatment may be helpful in most malignancies, your initial efforts should be focused on getting approval for the use of these drug combinations in the treatment of stage 4 cutaneous and ocular melanomas. 6. Dr. Arguello will assist you with all regulatory studies to get FDA approval for this new drug-based strategy to treat metastatic melanoma. Future studies and off-label uses of these drug combinations on other cancers will undoubtedly follow.
Clinical case of Susan B. This patient was staged and diagnosed at age 59 in Toronto, Ontario, Canada, as having a T3N2M0 grade 3 of 3 HER2-negative adenocarcinoma of the right breast that was positive for both estrogen and progesterone. She was treated with neoadjuvant chemotherapy followed by a modified radical mastectomy and 25 sessions of radiation. She continued taking letrozole for 2 years. Approximately 24 months after the operation, recurrence in the surgical scar was documented. A and B. By the time she entered our trial, at age 62, the cancer had consumed part of the soft tissue of the anterior chest wall, involving an area about 13 centimeters by 10 centimeters, and had invaded the left breast. C. Healing was evident 9 weeks after the initiation of atavistic chemotherapy, and D was complete after approximately 6 months of treatment. At the time of recurrence, a CT scan and MRI showed an irregular 5 cm by 4 cm metastatic tumor in the right parietal pleura next to the cardiophrenic angle, which also affected the pericardium and left pleura. E. Prior to trial entry, bilateral pleural effusions had developed, and a pleurodesis was performed in the right lung prior to atavistic chemotherapy. F. A chest X-ray taken 6 months after the initiation of atavistic chemotherapy shows both lungs inflated. Post-pleurodesis changes are also evident at the base of the right chest.
Hello, my name is Alice Brierly, and I live in Canada. In 2010, I was diagnosed with superficial spreading malignant melanoma. It started on the top of my right ear, metastasized to my neck, and eventually to my lungs. At first, I was treated at a neighboring hospital, but eventually sent to Princess Margaret Hospital, downtown Toronto, where they specialize in cancer. For 2 years, I took the traditional cancer treatments. No positive results. One day I happened to be at my family doctor's office for an unrelated reason, and I met a woman who was taking Dr. Argrello's Atavistic chemotherapy treatment. She showed me pictures. In 6 weeks, she had dramatic results. So I decided to take Dr. Frank's treatment. Almost 4 years it's been, and I am cancer-free. I'm still cancer-free. Many children, men, and women are dying unnecessarily from cancer, from traditional cancer treatments that are toxic, useless, and profitable. Thank you.
Clinical case of Alejandra G. This patient was staged and diagnosed in Torreon, Mexico at age 53. A stage T3N0M0 invasive grade 3 HER2-negative estrogen progesterone receptor positive adenocarcinoma of the left breast was found, and she was treated with neoadjuvant chemotherapy. After the 5th cycle, she chose to discontinue this treatment because of intolerance. After 12 months without formal treatment, the patient, now age 55, presented to our clinic with intense pain and shortness of breath. A. At the time of enrollment, the cancer had digested and replaced the entire left breast and was invading the anterior and lateral chest wall. B. After 1 month of atavistic chemotherapy, tumor vascularity had markedly decreased, and C. A complete clinical response was evident within 6 months of the initiation of therapy. D. At 20 months, marked fibrosis and retraction were evident. E. Radiological studies initially revealed tumor infiltration into all soft tissue structures up to the pleural cavity, with a massive pleural effusion. Evidence of resolution was documented after 6 months of treatment. F. A small loculated effusion that tested negative for malignant cells persisted in the left costodiaphragmatic angle. The patient also had a 3.2 centimeter liver metastasis that was no longer visible after 3 months of atavistic chemotherapy. Clinical case of Linda L. The patient was first seen at age 64 in Mississauga, Ontario, Canada. An infiltrative ductal carcinoma of the right breast was diagnosed and staged T1N0M0, and found to be negative for estrogen, progesterone, and HER2, triple-negative breast cancer. After a lumpectomy, the patient underwent conventional chemotherapy and radiation, but 12 months later she had local recurrence at the surgical scar, and a modified radical mastectomy of the right breast was performed. 2 months later, a second local recurrence at and around the surgical scar was detected, and palliative conventional chemotherapy was initiated. When the tumor continued to progress despite treatment, conventional chemotherapy was discontinued after 4 cycles. A. By the time the patient enrolled in our trial, at age 65, several raised islands of highly vascularized tumors had infiltrated the skin at and around the surgical scar and the left breast. B. 3 days after the initiation of atavistic chemotherapy, the tumors appeared pale and dry with small vesicles. C. 10 days later, the tumors had flattened out and superficial tissue had begun to peel off. D. 3 months later, the surrounding skin appeared to be near normal and scar tissue had developed.
In January 2014, I was diagnosed with metastatic melanoma, presenting with 4 brain tumors, one which had burst. I was treated at Vancouver General Hospital. In January 2014, they removed the burst brain tumor, and the 3 were inoperable. In talking with the oncologist there, we discovered that there were few treatment options for this cancer. We came home, and a good friend and neighbor gave us a reference to Frank's website. We looked it over, and we chatted with Frank and decided to get that sounded like the therapy for us. I took on the therapy and started in March 2014. And the scans in July 2014, I was tumor-free. Frank's provided me with who knows how many years more worth of life, and his treatment works. I think he's great.
Please note that this new drug-based approach requires different models of clinical trials, because the medications and strategies are not utilized in traditional cancer therapies. It is our duty and responsibility to change that. In other words, we cannot build the cancer treatment of tomorrow using yesterday's models for clinical trials. In closing, I hope you recognize the enormity of this opportunity and will take the appropriate steps to help bring this historical treatment to those individuals who continue to suffer needlessly.
I believe that this new approach for cancer treatment is going to change forever the way we understand and treat cancer in the world. It's going to save, hopefully, millions of lives and extend the life of many others. And also it's going to help us to save billions of dollars in medical costs.